Great, so a very different change of pace, and I'll be talking about how to prevent new invasive species, essentially. So, say we find a single alien animal in the wild, something like this red-eared slider turtle. Where did it come from? Is it just a lost, illegally kept pet like Fritz here, or is it just one of many in the area which are breeding and we have a new invasive species on our hands? We don't want to assume that a wild turtle is an escaped pet because a lack of action could let the population to grow, and then the cost of trying to eradicate that threat will grow with it. On the other hand, we don't want to spend a lot of money trying to eradicate a threat, which is just a single escaped pet. So what if we could figure out from the start if the animal's a lost pet or if it's from an established wild population? So this is what we had a look at, but first I'll introduce you to our test subject, which is the red-eared slider turtle. These guys were originally only found in America, but are now established on every continent except Antarctica because of their popularity as pets. What starts off as a very cute small animal very quickly grows very large and aggressive, and many people will then just discard their pet into their local pond. This might not be a problem as it might not survive. The problem is if someone else does the same thing. Two turtles could survive and breed, and as we've been hearing a lot of today, they can seriously threaten native species in agriculture by competition, predation, and by carrying disease. So we need a way to tell the difference between a discarded pet and a true wild animal to identify the threat of an establishing population as soon as possible. So we tried to do this using stable isotopes, which are heavier forms of elements whose abundance changes depending on the environment they're from, and they're very quick and easy to measure. As animals eat and drink, these isotopes accumulate within their tissues in abundances which reflect their diet. So if we look at how many stable isotopes are in a slow-growing tissue, we can get a long-term snapshot of where an animal's been and what it's been eating. So for example, a wild turtle will be exposed to the far more variable wild environment and eat mainly plants and a few fish. Meanwhile, a captive animal would probably be fed mainly the processed commercial pet food, which is very high in the heavier nitrogen isotope. So we looked at these stable isotope ratios in the keratin layer of the shell in 5 turtles, 2 seized from pet shops and 3 found in the wild. And what we found is that each turtle sits in its own distinct area no matter where we sampled on the shell. But more interestingly, we could draw a very clear line between the captive turtles and the wild turtles. So potentially, if we were to find a single turtle out in the wild, we could sample its shell for stable carbon and nitrogen isotopes. And depending on if it sits to the left or right of this line, we can quickly identify if it's from a wild population and we need to act immediately to start eradicating the threat, or if it's just a completely harmful lost turtle like Fritz. Thank you.